Thank you very much, Fraser, and good morning, everyone. I was asked to give an introductory talk on molecular machines working in solutions, so I apologize with some of you because for some of you I'm going to say, let's say, boring things, but I hope that after all, the presentation will be useful for the audience today. So let's start from a definition. A currently accepted definition of a molecular machine is a species, molecular-based species, where an energy input can cause the controlled large amplitude movement of some parts or some components of the system with respect to others. You can find these, let's say, sort of discussions in books and in several reviews published in the literature over the years. Now here I'm listing probably the very first one, the topic that appeared in 2000, and the very recent and comprehensive one published by the Lee Group in Chemical Reviews a few years ago. On the other hand, a molecule is an object. It's an object of a nanometer scale, and the molecules are the smallest natural objects, material objects, endowed with a shape. Everything that is simpler than a molecule is spherical, and another important thing about molecules is that they have, let's say, programmable properties in a way, whereas atoms have properties which have been determined by nature and we cannot modify them. By the way, does anyone recognize which molecule is this one? Any guess? It's caffeine, one of the most important molecules for the chemistry laboratory, of course. So we could say that when we want to make a molecular machine, the problem is to make a mechanical device at the extreme level of miniaturization in some regards. So the question is it possible to make operating machine at this level of miniaturization, or the topic is just good for science fiction, which has been for a number of years? Well, the most convincing answer to this question comes from the observation of the natural world. Living organisms, for example, including our body, are full of, let's say, molecular mechanisms, molecular machines. In this representation of the human body by a famous German artist several years ago, for example, muscles of the neck were represented with a little man pulling ropes. And there is some truth in this drawing, because indeed we do have ropes in our muscles. But these are nanoscale ropes made of molecules, molecular motors, particularly myosin and actin. And each myosin molecule can develop a very, very tiny force by burning a fuel. But thanks to a hierarchical, very precise and sophisticated organization of many of these machines in the muscle fibers, the collective operation can be, let's say, the operation of many molecular machines can be collected and produce an effect on the macroscopic scale. And this is an important thing to keep in mind. There are many examples of molecular machines beside myosin and the myosin that works in muscle. And the point is, are we expected, from an artificial point of view, to replicate such sophisticated systems? And of course not. We are not able to mimic in full the complexity of this system, but technology is telling us that we don't need to, let's say, replicate these type of devices. We just need to remember to learn from them, to learn how they work in order to produce our new, let's say, new prototypes. Nowadays it is possible, though it is, as I was saying, it's not possible to mimic completely the complexity of biomolecular motors. It's possible to prepare fully, let's say, artificial molecules of quite sophisticated structure. But from our, let's say, realistic point of view, what we can do, what we do every day in our laboratories, I believe, is to put together in a sort of rational way a certain number, a relatively small number, usually of molecular components, such that they can exhibit some kind of, let's say, non-trivial motion. Ideally, this motion should give rise to a function. In order to obtain, let's say, controlled and directional movements, we need to put energy into the system. We cannot simply rely on thermal motion, which is completely random, as Dean will explain us, I think, in a few minutes. And there are quite a few ways to deliver energy to a molecular system, and usually we can do it by causing a chemical reaction by using, let's say, fuels, or use light energy or electricity in a, let's say, sort of electrochemical setup. So the energy input is important, and this, of course, is a common property with respect to macroscopic machines. Also, macroscopic machines need energy to operate. Another point which is not, let's say, not of secondary importance for molecular machines is how we validate their operation, how we observe their operation. We need to exchange signals with the system, and usually these signals are of, let's say, consist of electromagnetic radiation, and there are a number of, let's say, analytical tools which are well developed in chemistry and material science to, let's say, to address this point. But according to my own experience, and I think it's probably experience that you also had in your research, in many cases, the difficult part is not to make the system, but to demonstrate that actually the machine works. A bit of historical, let's say, description, maybe. The birth of the field, the very first system that could be related to molecular machines were reported in the early 80s. This is a work taken from the group of Seiji Shinkai in Japan, and this group took azobenzene, which is a well-known molecule that is capable to change its shape in response to light stimulation. They attached receptors, molecular receptors or ionic receptors to azobenzene, and they were able to construct a sort of molecular tweezers for metal ions that were used to assist the passage, the transfer of metal ions across a liquid membrane by using light. But in this time, the concept, I mean, the idea of using a stimulus to cause a movement to produce a function was already there, but the concept of molecular machine was not yet, let's say, evident. It became evident over the years, and this is a News and Views piece taken from the issue of Nature. It's almost exactly 20 years ago, September 1999. The issue where the motors by Ben Feringa and Ross Kelly were published. So here people were starting to really pose the problem of making synthetic, completely artificial molecular actuators. Then the field has evolved over the years with a publication of a very large number of molecular machines and motors based on a, let's say, wide variety of molecular architectures, going from simple organic molecules to more sophisticated biomolecules like nucleic acids, and of course including mechanical interlocked molecules, which I will describe in more detail in a few minutes for reasons that we will see. And then the big thing happened in 2016, where the field was, let's say, consecrated. With the award of the maturity of the field was recognized with the award of the Nobel Prize to Jean-Pierre Sauvage, our hosts, Fraser Stoddart and Ben Feringa. This is the poster, very nice poster of the Nobel Prize 2016, where we can see this nice nano car. This is Feringa's nano car, overlapped with let's say the shape, the frame of a microscopic car. But this comparison can be useful, but there is also some risk in doing this, because it's true that molecular machines have several features in common with the macroscopic counterparts, but it's also true that the intimate operation mechanism is very different, and I'm not discussing this in detail because Dean Astumian will do it much better than me in a few minutes. But the only thing that I would like to emphasize is that I mean, it's not that the laws of physics are different from, let's say, in the macro scale and the nanoscale. The point is that the consequences of these laws can have different implications at two scales. So aspects that can be very important at the macroscopic scale are irrelevant at the molecular scale, and vice versa. So this we have to keep in mind when we design molecular machines. So now let's see a few examples. Of course, in 20 minutes I cannot give you, let's say, much details, but I selected a few examples to give you let's say, a clue, a feeling of the kind of reasoning that we do when we design and develop a molecular machine. So, mechanical interlocked molecules, they turned out to be very appealing candidates to make molecular machines for very simple reasons. First of all, you see this is a rotaxane, probably the archetype of mechanical interlocked molecules, where we have an axial component surrounded by a macrocycle. Because of the presence of these bulky groups at the extremity of the axial, the two components cannot dissociate. And so the mechanical bond confers stability to the system, but at the same point, there is no strong chemical interaction between the components here. There is no covalent bond. And so in these type of systems, there can be, let's say, a number of degrees of freedom of the components without, let's say, breaking the system apart, which can be exploited in terms of molecular machinery. And in fact, this type of motion, which consists of moving linearly the ring component along the axle, if the axle is sufficiently long, in a rotaxane, this is called molecular shuttling, is probably the most common realization of the molecular machine idea with artificial molecules, with rotaxane for sure. So, in very general terms, how should we reason if we want to design a controllable molecular shuttle? So a system where we can start from the ring on a position and then in a controllable way on demand, on command, move it in another direction. We should design a rotaxane where the axle contains two sides, two fragments, capable of attracting the ring by non-covalent interactions. So weak interactions that can be easily broken, okay? If these two sides are different, like here, these are colored with different colors, we can make that one of the two attracts the ring stronger than the other. And on, say, if we describe the situation with a simple potential energy diagram, it can be something like this. So we see that there are two barriers here, provided by the stopper, so that the ring motion is limited in this range. And we have two minima here corresponding to the interaction, to the attractive interaction between the ring and the axle. This is the preferred site because of the stronger interaction, so we have a deeper well. Of course, the ring can distribute between these two sites, and the precision in the positioning of the ring on this location depends ultimately on the energy difference between this minimum and this minimum. Okay, but we have means to, it's sufficient to have a few kcal per mole of difference in energy in order to have, let's say, the majority of the rings in a population located precisely on this side. Then we can perform a chemical reaction by which we remove the ability of the green station or the green site to interact with the ring. We do it by applying a stimulus. And at this point, we have generated a metastable state. So the ring is here, but there is no longer a reason for the ring to stay here. In fact, the system can relax and, let's say, can relax by moving the ring, by transferring the ring onto the now preferred blue station. The important thing to notice here is that the motion of the ring is not caused by the reaction, but the chemical reaction. With the chemical reaction, we are just telling the ring where to go. But the real kick for the ring is provided by thermal agitation, by Brownian motion. And again, I'm not going to more details because I'm sure Dean will discuss this point let's say, with more detail. Okay, so now we have reached another stable state, which can be subjected to another transformation. In particular, we can restore the properties of the primary site. If all the processes that we have used are reversible, this can be done. Again, we generate a metastable state, and Brownian motion helps the system to find, let's say, the new equilibrium and return to the initial state. Many examples of molecular shuttles that respond to this scheme have been published in the literature over the past 25 years. I would like to show you one of them, not only because it's taken from the chemistry of Fraser, but also because we have investigated this type of compounds together with him for a long time. And it turns out that this type of architecture is still widely used nowadays because it is simple, efficient, clean, and so on. So we have a rotaxane of this kind, where the ring is a crown ether, and the two sides are an ammonium unit, which is a hydrogen bonding donor, and a bipyridinium unit, which is an electron acceptor. The ring can interact with both stations because the ring can accept hydrogen bonds and is an electron-rich species, but this particular ring will prefer to give hydrogen bonding with the green station. So this is our starting point, and this can be demonstrated by a variety of techniques. So in solution, we start from this, let's say, this state here. We can add the base, a very clean process, one equivalent of base. We can deprotonate the ammonium station. It's transformed into an amine, so it is switched off. The ring doesn't want to stay here any longer, and we observe shuttling. At this point, the species is stable in basic environment. We can add an acid and go back quantitatively. And this cycle can be repeated many times on the same solution without degradation. This architecture was then used just I'm showing this also to emphasize the modularity of this approach. So the same stations were introduced in the arms of this trifurcated compound, you see the ammonium and the bipyridinium, which together with this tris-carbonyl component by self-assembly can give rise to a triply threaded architecture. And again, this process gives me the chance to emphasize another important thing. In our job, we rely extensively on self-assembly. Self-assembly means that we can make molecules that are programmed to recognize each other. We just mix them in solution. A solution is a fluid medium. At ambient temperature, the molecules can meet. And when they do, they associate together because the complex they form is more stable than the starting component. Of course, self-assembly is heavily exploited in nature. And it's the dream of any engineer, you know, to put all the parts on the table, shake them a little bit, go for a coffee, and when you are back, the assembly is mounted automatically. So this is actually what happens in our case. Then we can put stoppers and obtain a mechanical interlock system that was found to behave as a sort of molecular elevator because by changing the pH of the solution, we can move reversibly the red platform between two different levels on the rig. The same type of architecture can be used to make other topologies like this double interlocked rotaxane, which is called in our jargon C2 daisy chain. And this is interesting because using the same concept of molecular shuttling that I just described you can make a molecule that expands and contracts, so a molecule that can change its length reversibly in response to stimuli, which in this case are chemical stimuli, but you can make these systems operating by light stimulation, for example, or also electrochemically. The molecular elevator was also featured on the Nobel poster. We can also make molecular shuttles that use light, sunlight particularly, to operate. That was a very satisfactory and rewarding work, again performed together with Fraser several years ago. The rotaxane is a quite complicated species shown here, containing several moduli. All these parts are inserted in a specific position for a specific reason on the basis of specific properties. I don't have time to discuss the mechanism, but to make a very long story short with this animation, in this compound, absorption of a photon from, let's say, by a photosensitizer by a specific part of the molecule causes the transfer of an electron on that part here, which changes the relative affinity of the two stations for the ring. And so overall, we have that light can cause back and forth shuttling of the ring in the rotaxane. And the process can continue automatically, autonomously, as we say, as long as you leave the solution with rotaxane on the window, under sunlight. So the process can continue, let's say, without any other intervention. Can we do something, let's say, useful or prospectively useful with molecular shuttles? Well, an obvious step forward is to use the change in distance between the subcomponents of the system to control a distance-dependent process. And the thing that we made, of course, there are many options, but the thing that we made was to attach to the ring and to the axle two persistent radicals, which are, let's say, which are endowed with a magnetic dipole. And if they are close enough, their magnetic dipole can interact, and you can see change in the EPR, in the electronic paramagnetic spectrum. And if you change the conditions, you can, let's say, move the two radicals apart, and this interaction disappears. So we can mechanically switch on and off a magnetic interaction in this system. Another thing that the proof of principle, of course, we did was to exploit the shuttling to move a cargo. To do so, I mean, from a design point of view, you need to functionalize the moving ring with something to which you can dock a molecular substrate with a connection that you can control with the stimulus, which should be independent from the stimulus useful for moving the ring. And this is the most difficult functional requirement to meet, but this is something that can be done. We used pH changes to move the ring, as we have already seen, and we used light to unload the cargo once the ring has been moved. in this state. So we see that we start from this position. The ring can be moved by adding a base, and then upon light irradiation, you can, let's say, detach the ruthenium complex, which is then transported. Of course, the whole thing occurs in fluid solution, and so we are not transporting anything, of course, but the functional requirements are, let's say, are met in this system. Other things that we did, I have no time to discuss this in detail, but with molecular shuttles, you can modulate the acidity of a site by electrochemical modulation of a remote site in the molecule. And very recently, we used molecular shuttling to play games regarding chirality arising from the mechanical bond, which will be discussed, I'm sure, extensively by Steve Goldup. Steve, where are you? Steve Goldup tomorrow. Other very interesting, very, let's say, challenging and sophisticated things that can be done by molecular machines, not only mechanical interlock molecules, but also other type of molecular machines in solution, has to do with catalysis, with, let's say, controllable catalysis and programmed chemical synthesis. Many very interesting, very advanced studies were reported by the group of David Lee, but I have no time to discuss this. I need to get to the conclusion quickly. Before I conclude, in recent times we have been interested a lot in the development of a system of this kind, where we can cause the unidirectional motion of one component with respect to the other, something that you could change, that you could name molecular pumps. Molecular pumps can be useful for other, let's say, developments, like to make rotary motors or linear motors, or maybe one day even for creating gradients by using molecular machines. This is what we did in our case. Our pump works under the action of light in an autonomous way. You see that we were not much brave in the title of the paper, but fortunately ED Civic in the News and Views piece was more straightforward. What we made is a light-driven molecular pump. A few months later, Fraser came with this very successful and elegant design, and in that case, Fraser was more straightforward to emphasize that, in fact, they had made an artificial molecular pump. Okay, to conclude, let me, let's say, summarize a few concepts on molecular machines operating in solution. So why the solution? Well, studies in solution have been instrumental for the development of the field of molecular machine. There is no doubt about this. It's still important to study molecular machines in solution. Molecular machines often are quite sophisticated systems, and solution provides a simple homogeneous environment. We know very well how to characterize solutions, species in solution, and so it's good. Maybe engineers would say that solution is good for prototyping molecular machines, not to study new systems, to understand better the mechanism by which they operate. You can use solution properties to tune the behavior of molecular machine. This is a pro, but can also be a con, as we will see. And remember, in view of also potential applications, that there are interesting applications that can be performed in solution, catalysis, synthesis, but also remember that biological systems operate in solution. The cons, the main problem of a solution is that the solution is an intrinsically disordered state. Molecules are floating. If you need an application where you need to fix the molecules in a space or to arrange them together in order to, for example, to collect, let's say, to exploit collective behavior, then the solution is not a good choice. Another problem is that in many cases, molecular machines rely on solvent to operate, because a molecular machine is a molecule, and solvent is also made of molecules. So this molecule very frequently intervene directly in the operation mechanism. If there is no solvent, the machine doesn't work anymore. Machines based on hydrophobic effects, for example, if there is no water, there's no hydrophobic effect. And this is a problem. So I think we need, for the future, to develop more and more examples of molecular machines that are more tolerant with regard to the environment. And I think one strength of the motor, of the rotary motor of Benferinga, is that it can operate in a variety of environments. Okay, so I leave you with a picture of my city, and I thank Fraser and Melissa for the invitation, and all of you for your attention. Thank you.
Grazie.
Prego.
So most of the devices that you showed, they have either translational or rotational motion. To build more complex functions, probably it's good to combine this, like in the industrial machines. Has anyone tried to do it for molecular machines?
Yeah, this is a very good point. A challenge for the future is to combine molecular motors, for example, with passive elements. No? In a macroscopic machine, usually you have a motor component which transforms the input energy into a force, and then this force is transmitted to gears, for example. This can be done for molecular machine. It's difficult, but there are studies which go in this direction, so it's completely true. So with the rotary motion, you can actually use this approach to transform and process the motion and to convert into some, say, more sophisticated things.
So I'm thinking of like applications of some of these things you developed, and what stuck out the most to me were these pumps and maybe pumping things into the cell. There's a lot of challenges getting into the membrane, and
Absolutely.
maybe the first one's toxicity. Have you ever just fed these to cells and see if they survive?
Good point. I'm sure that if we provide cells with the molecules that we normally study, the cell would die almost immediately. They are not designed to be biocompatible, in most cases at least. But apart from toxicity, of course, I mean, toxicity is an aspect that can be double-sided, because actually one purpose for a molecular machine, if you want to use it as a drug, could be to kill the cell, in fact. No. The problem to, let's say, to have molecular machines working in cell membranes, for example, is, as I said, let's say, the fact that the operation of the machine is maintained in the cell membrane, the fact that you can embed the machine, for example, in the case of the pumps, to the ability to embed the machine vectorially in the membrane, so in an oriented way. So at some point, natural systems are extremely well organized, and this is, I think, where we need to make the most progress.
So you said that there's certain disadvantages to having these in solution. I would say these are advantages, because ideally what you want is distributed functionality where things come together for a specific function. Having done that, they move on to a different place and do other things.
Yeah.
And you can only do this in solution.
Yeah, I'm not saying that the solution is the evil. No, there are many interesting things that can be done in solution. Biomolecular machines, after all, work in solution, but they work in very organized, compartmentalized, structured solutions. Because, for example, if you want motor proteins transport substrates, you know, moving in a solution, but they are always connected to a tract. If they disconnect at some point, mixing in the solution will nullify the effect of transport. So I agree with you, but on some other regards, the fact that you have such a disordered environment can be a problem.
All right, I think we move on because time is important. But before we do, thanks a lot, Alberto.
Thank you. Thank you. Thank you very much.